Hi, today we are going to take up cerebrospinal fluid and its functions as a part of the blip. In today's topic, we are going to see the formation, circulation, and how it is reabsorbed into the blood. So, to introduce this topic, CSF, it's clear colorless fluid, and it is around 150 ml in volume. That is there in the ventricles and all around the brain and spinal cord and in the central canal. Then, as you know, this is almost not having any blood cells and a little amount of protein. To introduce its rate of formation and flow and absorption, it will be around 550 ml per day is produced, flown through various chambers and absorbed. That means it is roughly 3 to 4 times of the original volume. That means we are replacing it three times at least. The 150 ml is replaced 3 times at least with fresh fluid. Then measuring its pressure, CSF pressure, and performing cell counts, analyzing levels of various biochemical constituents can be accomplished by performing spinal tap. And we have some clinical correlations to the CSF analysis. To start with the formation of CSF, 70% of the CSF is formed by choroid plexus, which is present in the lateral ventricles, third ventricle, and fourth ventricle. Remaining is by various other structures like ependymal lining of the ventricular system and other components will also produce CSF. Majority is 70% is by choroid plexus, which is nothing but a cauliflower-like growth into the ventricles covered with the lining, epithelial lining. And this is in this picture you can see. On either side we have ventricles. In the center we have the third ventricle. These two are lateral ventricles. I can see interventricular foramen is connecting lateral to third ventricle. The third to fourth is connected by mesencephalic aqueduct or aqueduct of Sylvius. Then in the fourth ventricle you can see through two lateral apertures, one median aperture, ultimately to make the CSF all around the brain and into the central canal of the spinal cord. And absorption is by arachnoid villi. Arachnoid villi are nothing but the protrusion of arachnoid membrane into the central venous sinuses and other sinuses. That is a major part which is reabsorbing. Then minor parts include capillaries and veins of the CNS can also reabsorb CSF. Then functions, three functions. To start with, always protection in terms of cushion, shock absorb by doing modulation of the pressure changes. That is the first and foremost function of the cerebrospinal fluid or CSF. Then second is it is also serving as a chemical buffer to maintain constant ionic environment around the brain. Then it serves as a transport medium for nutrients, metabolites, and endocrine substances also. So this is the third function of the cerebrospinal fluid. Now we'll take a 1 minute on blood-brain barrier, which is a physiological barrier for substances from blood to the brain tissue or brain parenchyma. This barrier is important to maintain or to preserve a stable environment for the CNS, which is the key tissue in the body. Now, how the blood capillaries are different when compared to general capillary. So, the blood capillaries in the brain are having tight junctions so that nothing leaks out into the brain tissue. Then reduced vesicular transport or pinocytosis, and foremost, last but the most important point is the food processes of astrocytes. I'll show one diagram to understand that. You can see this is one astrocyte, and the food processes are ultimately covering this capillary all around so that nothing leaks out. This is extra reinforcement so that nothing will leak out of the capillary and enter the brain tissue or brain parenchyma. Then the key observations on the blood-brain barrier are this barrier is allowing lipid-soluble substances without any resistance. That means they will pass out with so much of ease and free. That means carbon dioxide, oxygen, alcohol, which are all lipid-soluble substances, they can pass through the barrier with ease. Then one more observation is negatively charged molecules cross more freely when compared to the positively charged ones. Then one more applied point is during injury and inflammation, the resistance of the blood-brain barrier is decreased. That is useful in such a way that it allows antibiotics that are used in the treatment. So with this we have covered two important topics which are so small but they are very much crucial in Viva also. See you in the next video.